home who've just tuned in, we're on Santa Lucia Bank off Morro Bay, off California. And right now we're doing a quantitative transect. That means we try to keep the height above bottom for the ROV and the speed relatively steady, and then go a distance of about 200 meters. It makes a transect for us. And that allows us to get quantitative estimates of species abundance during by counting the animals in the photograph. And then, because we standardize how we do the transects, we can compare it to other areas in the region and other areas coastwide.
Kim, you want to look a little down on the camera? I'll just, we'll try to maintain a little bit higher like that. Yeah, a little tighter. That's great. Thank you.
And we're seeing scattered sponges and those white things that have like tentacles out are actually sea cucumbers. So, and then Liponema, an anemone, purple anemone going by, little barrel sponge there at the bottom of the screen, and a solitary coral whose name I can never say. Erica, hit me with that name again.
Heteropolypus.
Why can't I say that? I think they look like something out of a cartoon that has toadstools. There's one right going into the center of the screen now. You can see the pink polyps out on that feeding. You can draw those polyps in, and then it really looks like a toadstool, just white with pink polka dots. That stick-looking thing out there in the distance at the top of the screen is a coral, a deep sea coral. And that a little deep sea soul there in the center of the screen, Erica, going off to the right?
Looks like, yep.
Maybe not. Thomas saying the same mushroom coral instead of struggling with heteropolypus. There, I said it once. Another mushroom coral, heteropolypus. Ooh, said it twice now. Is that a scarlet king crab there, Erica?
It is. It's either a Lithodes or Neolithodes. You really get the feeling that we're going off the edge when you look out to the left.
Yeah,
Oxorid coral.
looks like we'll be flattening out probably in another 60 meters. So it should be just the plane.
All right, friends, I believe it is that time of the morning. EB time.
Yeah, we've gone from lots of sponges to
You gotta come down a little.
not very
Gotcha.
many. One little IKEA lamp. Couple of Thanea in that view. Oh, our first CPAN of the evening, the morning, I guess.
Maybe a sign of things to come.
Yeah, if we're heading to flats, we will definitely be switching over from corals that like rocky substrate to corals that like muddy substrate.
Say that again.
Another CPAN. Seeing more of those.
Kind of see why they call them sea pens. They sort of look like a plume pen. Deep sea sole there, the bottom of the screen going by.
The pattern's so much brighter on the smaller ones.
Yeah, I was just thinking that. And I thought maybe it was my imagination, but I guess not. Have you told the story that you'd like that pattern as wallpaper in your house?
Not yet. But there are companies now that make clothing in different patterns. You can get whale shark spots or tiger shark stripes. So I might have them add deep sea sole spots to their repertoire.
As long as it's imitation.
Right.
Yeah. Wow, you can really see the current here. That sea pen just waving in the current.
Last couple sponges clinging to any rock. Before we head out onto these flat areas.
It does look like coarse sand here, doesn't it?
Yeah, a bit, yeah.
Maybe even gravel. The next size up on top.
What's the finest? It's
So,
silt?
Mud. Yeah.
Mud, yeah.
Mud. I don't have my key in front of me, but they have it down to millimeters. If the particle is so many millimeters by so many millimeters, it's considered sand.
it's a little hard to do those descriptions when you're just looking at photographs. Erica does a lot of that. So I guess, Erica, you get kind of a feel for it, don't you?
Seems like Eric has a feel for everything down here. All the experience.
Sand 70 by 70.
I think 7.
Oh, we're getting into some brittle stars, which we haven't seen. For a while.
Roger.
See if we start seeing more fish out on this open plain.
Sorry?
Oh, I'm just talking. Just I said it'd be interesting if we see more fish out on this open plain, more flatfish.
No, I mean after the, yeah.
Another thorny head. Commercial fishermen call them hardheads, right? Or idiot fish, some of them.
Bucket
It's
heads?
another name. The fish. Bucket heads, is that it? Yeah.
That's what Erica said.
For the larger short spine,
Oh.
yeah. Yeah, I mean, they have lots of names. And this I would definitely call gravelly mud. I'm looking for that article to give some of the measurements. Because it is sometimes comical when you're supposed to do it from a video or a picture.
Right. Can imagine.
Is that a 6 centimeter cobble or a 5 centimeter cobble?
Both.
So for the people at home, we use the lasers. You can see those two dots there to give us a sense of size. Those are 10 centimeters apart. And then with the equipment that Erica and I also use, which is an autonomous underwater vehicle, we have stereo, not video, but stereo still cameras. And because they're stereo, that allows us to take measurements fairly accurately, but probably not accurately enough to tell the difference between sand and fine gravel.
Need a really good calibration.
Yeah. Well, we do that. We
Yeah,
calibrate the cameras before every cruise, and usually do that in a swimming pool, actually.
Big chessboard.
Pardon?
With a big chessboard
Yeah,
or
we
something?
do.
Yeah.
Yep, a big chessboard, a big black and white target. And then we always do it after the cruise again. So that gives us calibrated cameras so we can take measurements. But it's tedious because let's say you're going to measure a fish. You have up on the screen the stereo photographs, and you have to by hand click on the head and click on the tail in one photograph, and then move over to the other half of the stereo, click on the head and the tail, and try to do that as accurately as possible.
Well, it's not that different than some of the microscope works where you do it in the software. I think I've done that with the larval fish. So actually we have, we've come 200 meters right now, so we can call this the end of the transect, T6.
Sounds good. And now,
so
you can go pretty quickly.
Great, yeah, so we'll step us up. 0.5 knots. Yeah, let me get it. It's not gonna be that different. Like 275. 275. Yeah, let's do that. And 0.5 knots. Okay.
So 0.5 knots is that's kind of really stepping quickly with this ROV, isn't it? What's your sort of max speed you're comfortable with?
You're not on SPM.
Yeah, 0.5 knots will be fine for us, and depending on the current and if we get blown off and things, but yeah, we can give it a go.
When we're in the blue water, we've done about a knot.
Yeah,
that
we're in blue water.
Yeah, when there's nothing to run into.
Right.
Give me one phase 275, please.
So I've found my list, and the definition of gravel is 4 millimeters to 2 centimeters.
4 millimeters to 2 centimeters.
And then it becomes a pebble at 2 centimeters to 6.5 centimeters.
Oh, man. Sediment people are very nitty-gritty about their details.
Yeah.
Nitty-gritty, very funny.
Sediment puns. I mean, I understand why it's important. It just might be seem arbitrary or frustrating to have to count all that. Yeah, I already did. Is that all right? 275. I'm going to do 50-meter steps. Maybe more.
So we're going to move across this flatter area and try to find another upslope on a different section of the bank, and hopefully see more corals and sponges there. We're just going to try to zoom through this flat area.
Yep, this flat area is got more sea pens and more brittle stars. And there's another little deep sea sole. We're kind of out of the sponges now because there's nothing for them to hang on to. And just as I say that, there's a little barrel. Nope. Little coral. Is that a black coral we just went over, Erica?
I did not see it. That one, I think they're plexurids.
Sorry. Science, the only thing about going 0.5 is that we won't be able to really stop and zoom in, especially with this current.
No, that's okay. If we we are happy with that. You can see a brittle star there hanging on to a C-pen. There's
Thanks, Ben, for the
a
catch.
sponge who's found a rock.
Yeah. Oh, yeah. Muddy bottoms.
And the Dover sole? No, deep sea, deep sea sole. Surprised we're not seeing more Dover in here. Does that surprise you, Erica?
Yeah, it looks pretty flat, right? Yeah, this current's good down here, man.
Are you going with it or against your?
We're going a bit
Pushing
kind of
you.
orthogonal and against it.
Is it coming from the north, it looks like?
Yeah, it's
To
like
your,
go ahead, sorry.
to starboard, right?
Yeah, it's moving from right to left, so it's, yeah, like
I wonder, because we're
north,
like in
northeast.
this like little channel in the bank. So we're going from one side of it to the other, and it looks kind of like it's coming down that channel, or at least that's the direction it is.
Yeah, I'd buy that.
Trying
From
to sell
IKEA.
it.
Erica, what was that fish that just went out of the screen that I missed?
Might have been a Basilaga. Basilaga is a deep sea smelt.
The larvae of those are very interesting. Look very different than the adults.
So science, this is kind of the movement of us without any forward thrust. So we're just moving with the current right now. As you guys were asking about it earlier.
Too bad it's the wrong direction.
Oh yeah, we'd be so cruising. Sailboat.
that a sponge? The little spiral thing going on? Or no?
Not seeing spiral.
Yeah.
It's
like,
funny,
yeah.
every rock has something stuck on it.
Clinging for dear life.
Right. Any little bit of hard substrate.
Things down here are definitely opportunistic.
So that fish we saw, the bathylaggid, when I was saying that the larvae, the babies of those fish look very different than the adults. Bathylaggid larvae have their eyes that are stuck out on long stalks and look very, very different than the adults. If we see another one, you can see that the adults look nothing like the larvae.
Now you're going to make me Google it.
Yeah. Bathylaga is what? Is it a stomiid or what is that?
Yeah, they're generally called deep sea smelts.
Oh, smelt. Yeah.
Oh, that is weird.
Isn't that odd?
That's very alien. Is that unique to this group?
Yeah, I don't think I know of any other larvae. There may be some that have those stalked eyes, but they're the ones that I know for sure have stalked eyes. I have to look it up here. Something called a black dragonfish has stalked eyes too. Probably is a way of seeing better, enabling them to improve their feeding efficiency and possibly predator avoidance as well. The black dragonfish is an idiocanthus, so a very different species. And their eye stalks, as I remember, are much, much longer in some cases.
Yeah, the larval fish are so different sometimes. I just, my old lab, we had some, ichthyoplankton experts who were so good at the larval fish ID, and you'd show them an adult, herring, and they'd be like, what's that? So specialized, such a different body form.
Yeah, I did a lot of my early work and my graduate work on larval fish. And so
that's cool.
When I'm talking to Erica, I say, and I can't quite get the adult fish ID down. I said, yeah, but if you gave me one that was less than a centimeter, I could probably tell you what it was.
Yeah. Exactly.
They do look very different. I mean, one of the best examples, of course, is the larvae of flatfish.
Right.
Look like a regular fish with one eye on either side of the head. And as they mature, the eye migrates over the top of the head. So both eyes are on one side of the fish. And so really, when you see a flatfish, it's lying on one side only. And its eye has rotated over. One of the eyes is rotated over. They call that transformation. that stage. And that's when they go from being a larval fish to a juvenile fish, really, or a teenager in the fish world.
I've seen a couple of different sea pens. I've seen Umbellula and also Anthoptylum. There's another Anthoptylum going out the bottom now, along with the plumes that we were seeing earlier.
Lots of brittle stars on that sea pen. They're probably not feeding on the sea pen. They're just getting up higher in the water column. Get better feeding if you're not in that close to the bottom layer. Current speed is higher. Just get a little bit off the bottom. And one rock, so got some sponges on it, a little barrel sponge. So you can see how it looks like there's sediment around the outside of that barrel, and then it's clean white on the inside. A Dover cell, Erica, going by?
Yes.
And those barrel sponges, they have long spicules, sort of the the glassy part of the sponge that gives it some structure. And those long stipules catch the sediment. So they look dirty on the outside, but they'll be clean white on the inside. Pretty sea pen. So how much longer on this flat part do you think before we get back into the rocky area?
Yeah, we're about halfway until we start coming up slope a little gradually. So
Okay.
I can calculate it for you. I think it was,
About another half hour?
yeah, probably something like that. From the other side, it was going to be an hour and 20 at 5.5 knots. So let's see. To the target
Okay.
itself, it will be like 50 minutes, but we might start going up slope maybe 20 minutes before that.
Yeah. Oh, what is that?
Did you guys still have shell hash on your collections list? Or do you guys want to
Yeah.
keep
I
going?
think
Earlier?
yeah.
Could we slow here if we can? I know you're going pretty fast to slow,
You want to stop the ship?
but Erica, do we still have shell hash on there?
They made a collection earlier.
Oh, okay. Roger that.
Oh.
We don't need
Can
that.
we zoom in on that hash? Oops,
Sure.
never mind.
No, that's good. All right, Marley, go ahead. Doesn't
Huh. Interesting.
sorry. A little bouncy. You want to come
That
wide there,
doesn't
Marley?
doesn't look like shells.
No.
No, it doesn't. It,
It's almost carbonate.
yeah. Wonder if it is some sort of a older seep.
All right, Marley, come wide,
Interesting.
please.
Thank you.
All right, Ben, go ahead and start us
Yeah, I
back
want
up.
to go.
Roger that. D. Roger.
They're dorsal, making some way there. They're funny the way they swim with that undulation.
And you want to square up there on the
Oh, yep.
ROV nav?
So,
Thank you.
So we're making some way here, going pretty quickly across this flat area. Just for people at home, we're on Santa Lucia Bank off Southern California. We've been in some rocky areas with lots of corals and sponges, and now we're going over some flat areas that is more gravelly. We're not seeing as many corals and sponges, even though we are seeing corals called sea pens. And there you see one there with a brittle star wrapped around it. I'm trying to get up into a good area to feed. This area of Santa Lucia Bank has not really been explored much. So this is why we're here, trying to get some information in an area that we know pretty little about. We don't know much about the biology, and in addition, the geology has turned out to be quite interesting. Doesn't, of course, if you're tuning in now and you're looking at this sandy area, you're thinking it's not very interesting geology here. The sea pen on the bottom, Amphiphyllum, I think it is, probably Erica, am I right?
That's correct.
And Paralithodes or Lithodes crab we want to buy there. And we have a rock with a coral on it and a, oh, no, sorry, an anemone.
what's that ahead?
No, it's
Long.
like kelp.
Yeah, up top. Debris, some sort. Or kelp?
Yeah,
Kelp.
yeah, so one of the things that's interesting about the deep sea for people at home is we're out of the zone where there's photosynthesis. So most of these animals depend on what falls from the surface for food. So you can see that that's a piece of kelp that's come from way farther inshore and drifted down. And that's a food source. Most of the food that drifts down is in the dead plankton or small planktons that make it down this far, small little inverts, but also marine snow, which is just organic material drifting down from the surface where there is high productivity because there's sunlight and things can photosynthesize. But that's why you see interesting feeding adaptations in the deep sea. You see things that have, oh, there's another piece of kelp. You see things that have big mouths. So when they come upon food, they make a feast of it. And then they make one big meal and take a little while to digest that. So they want to make sure they eat as much as they can when the buffet is there. So big mouths to get whatever's around. Small size is sometimes an adaptation for the deep sea because there's not much food. You want to probably keep your size small, except there are some examples of extremely large animals in the deep sea. And that can be an adaptation to avoid predation or to allow them to have huge meals and digest them. These adaptations down here in the dark where there is no light, not only can you not photosynthesize, but there's no light for vision. So a lot of the organisms in the deep sea have adaptations to, they have photophores, they have ways of using, in generating their own light. A lot of times that has to do with having bacteria that actually generate the light for them. They have big eyes, so they can capture what little light there is. There's also lots of adaptations to the pressure in the deep sea.
What's the distance to the...
Yeah, we still have 0.3 nautical miles, which
Roger.
is like 500 meters.
Silver Soul.
Roger that.
Hoping that little bump before it is interesting.
Sorry?
Hoping this little bump before it is interesting.
Yeah. Seems like we're going to set up Bob's watch to have a good upward slope.
Can't have it all.
Hi, Di. Good morning. Another one of our scientists ashore has joined the room, and I think she'll be coming on. No, it's Lizzie and Abby. I thought it was Di and Abby coming on after us at the shift change. But hopefully Di will stick around and help us ID things.
We also have Meredith back on, Meredith Everett, who's our coral expert. She said we zipped over a Tassia, sorry, a little while back. I missed it. No, little snailfish floating in the water column. We're saying how larval fish look different than the adults many times. In the case of snailfish, they actually look a lot like the adults. So they're little tiny larval fish with the same chubby cheeks. So we're zipping along here, about 0.5. It's a little hard for us to stop at this point because we're trying to make some distance here to get in some of the rockier areas. We did do a transect in this flatter area though. Erica, is Tassia on our collection list, do you know?
Yeah, we could, if you want, we could do a transect here too again. It just, if you don't mind it being a little speedy, but just because we have, we're essentially doing that.
Yeah. Well, why don't we mark it as a transect so at least people will look at this, and if it's a little speedy for them, they can probably make do. Okay, yes,
And yes,
Tom is saying it's good for a transect here because it's so flat. So
Great.
let's